A book entitled Learning the Brainstem, or LTB for short, exists in the marketplace. Why not have really short videos to capture the book's main points? The voice you hear is AI generated. Reason being, we use AI to facilitate translation into multiple languages. This video, LTB4, continues our discussion from LTB3. Our location is still the high midbrain. Given that we orient our brainstem as we visualize it in a radiographic image, the hemi-brainstem to the right side of the slide is the left midbrain, and the hemi-brainstem to the left side of the slide is the right midbrain. We will rely on the normal bilateral symmetry of structures in the brainstem, so that when we identify structures on one side, for example in the green shaded area in the right midbrain, we can see the same structures in the contralateral midbrain without superimposing anything. In the green shaded area are the right cerebral peduncle, which is dark, and right substantia nigra. It is a nucleus rather than a fiber tract. It is pale, and we will abbreviate it as SN. I want us to be oriented in three-dimensional space, so in this slide I provide a coronal rather than an axial section. The red arrow points to a lens-shaped right subthalamic nucleus of Lewis, superior both to SN, as indicated by the grey arrow, and to the cerebral peduncle on that side. The red bar in the left hemibrain passes from internal capsule superiorly into the left cerebral peduncle, inferiorly and ventrally, but the peduncle and the capsule are contiguous. So, this entire axial section is inferior to the subthalamic nucleus, which we don't visualize at the present level. As just said, ventrally there is a dark cerebral peduncle, and immediately dorsal to it is the SN. There are parts or portions of both structures. Knowing about the different parts deepens your knowledge base. There is one other comment with respect to a bit of midbrain medial to the green shaded area, circled in red, along the ventral surface facing the interpeduncular fossa. It is called the ventral tegmental area, which we will revisit as we move lower in the midbrain, below the current level. One can divide the cerebral peduncle roughly into thirds, as indicated by 3 blue dots. Medially, there is a frontopontine tract, frontal lobe to pontine nuclei. The central third contains corticospinal fibers, cortex to spinal cord. A lateral third contains a temporopontine tract, temporal lobe to pontine nuclei. The idea that the cerebral peduncle only contains corticospinal fibers is just wrong. The blue dot now sits in SN, which consists of two parts. First, there is a portion called the SN pars compacta with cells that contain neuromelanin. The SN pars compacta is best visualized with a cell stain or in fresh autopsy specimens. The grey arrows point to collections of pars compacta neurons that contain neuromelanin as visualized by cell stain to the left and in a gross pathological specimen to the right. In our axial section, SN appears uniformly pale, as if to remind us that there is also a pars reticulata of SN, as opposed to the SN pars compacta. Reticulata neurons behave physiologically like medial globus pallidus neurons. The SN pars reticulata is an output nucleus of the basal ganglia. Its output is directed to thalamic nuclei, but not the ones visualized in this slide. The thalamic nuclei that we do visualize, shaded in green, include the medial and lateral geniculate bodies, but not just those two. Let's place dots on structures. First, medial geniculate body. Next, the lateral geniculate body. Note that dark fibers of the optic tract enter at the concave surface, as indicated by the red arrow. Optic radiations, on the other hand, leave from the convex surface. Finally, there is a bit of pulvina. At the tip of the red arrow, notice a tinge of white matter that represents the brachium of the superior colliculus, which we will address more fully in LTB6. Now the green shaded area encircles the red nucleus. It is called red because in fresh brain dissections it looks pink or iron reddish. But it is hard not to notice that the nearly circular red nucleus itself is surrounded by white matter, and dorsal to the green shade, lateral to the central grey, and extending as far laterally as the medial geniculate body, there is a heterogeneously light and dark area. What structures occupy the space? Let me start just at the midline. White matter does indeed encircle the nucleus, so that you can follow the white matter around. We follow the white matter with two more red arrows. The fibers are mainly those of a cerebellum to thalamus via red nucleus projection. That is, fibers from contralateral deep cerebellar nuclei will have already crossed the midline at the level of the red nucleus. But the fibers around the red nucleus do not constitute just one fiber tract. Other fibers wrap around the red nucleus. Lateral to the red nucleus, fibers that arise from medial globus pallidus loop on their way to thalamus, as was described by Auguste Henri Forel in the 19th century. The red arrow points to what Forel called an H field. H is short for Haube in German, a cap like a baseball cap. If you think about white matter a bit more dorsally, it helps to remember that the ventral thalamus is just superior to where we are. And the lateral midbrain tegmentum, where the red arrow points now, will by necessity contain the ascending fibers of sensory tracts from spinal cord and trigeminal sensory nucleus, as well as ascending auditory projections. We already have some information regarding the area inside the red rectangle, whose medial edge sits next to the posterior commissure. Inside the rectangle is pretectum, and there are nuclei there, among them a pretectal nucleus and a nucleus of the posterior commissure. Previously, we discussed nuclei within the central gray around the cerebral aqueduct and in the rostral interstitial medial longitudinal fasciculus also at this level. Fibers from pretectal nuclei cross in the posterior commissure, which is a landmark that we soon will no longer have, as we pass to an axial level below the posterior commissure in LTB 5. LTB 4 worked through quite a lot. Thanks for watching, and remember what I have said about the LTB series as a whole. We want to satisfy the curiosity of many kinds of students, from beginners to those who teach. And regarding the latter, I promise you, they are still learning.